I'm Willow from Center for Informatics, Harvard Institute of Technology. Here, I'm very delighted to introduce our new work on the indexing of large amount of genome sequences, the branch-based BWT constructor. Usually, we shout at deBWT. My presentation divides into 5 parts. And at first, let me introduce some background and the motivations about the work. The number of genome sequences is rapidly increasing. To this June, the number of sequences recorded in GenBank has been over 194 million. And more than that, in the future, the number will continuously increase. Last year, Stefan et al. Published a study on the growth of DNA. It predicts that in the next 5 to 10 years, there will be 100 million to 1 billion human individuals genomes being sequenced, which is an amazing number. Facing to such a great amount of genome sequences, there is a very important problem, that is how to well organize and index the large amount of genome sequences to facilitate future genomic studies. Burrows-Wheeler transformation, usually short as BWT, is a feasible choice because of the linear complexity in retrieval and storage. It has been widely applied in genome indexing, sequence alignment, genome compression, genome assembly, sequencing error correction, and so on. So many applications. And we believe that it will go on to play an important role in the future. However, a non-negligible problem is that it is non-trivial to construct BWT for many genomes. Briefly, given input sequence, BWT is the concatenation of the previous characters corresponding to the sorted suffix. Here is a toy example. And in essence, it needs to determine the lexicographical order of the suffix to construct BWT. This task is not easy, especially for those suffixes with long common prefix. The task is even more difficult for many similar genomes, such as large amount of human individual genomes, because there will be many repetitive sequences, so that a lot of suffixes would have long common prefix. So efforts have been made in recent years for BWT construction. Many proposed methods are in an incremental construction approach. In 2007, Hong et al. Proposed important work in this field. Other important works also include, but not limited to, BWT disk, PCR, BWT2, and PairBWT, and so, etc. This work mainly takes advantage of the error mapping property of BWT. With this property, it's feasible to partition the input sequence into blocks and construct BWT blockwise. These methods are fast and lightweight. However, they're hard to implement parallelization, as the operation on a new block depends on the results of the process blocks. This could be a bottleneck in future. So, since it's hard to fully take advantage of the modern high-performance computers to handle a large amount of genomes. There are also non-incremental approaches, such as the work proposed by Karkanian, based on different cover sample. Further, okay. Okay. And CX1 is also good work, which constructs BWT of many short sequencing rates in very high speed. It mainly uses GPU-accelerated relative sorting, which fully utilizes the parallel computing ability of GPU. Non-incremental approaches are more easier to implement parallelization. However, their ability of handling suffixes with long common prefix could be not as good as that of the incremental approaches. And they are usually not efficient enough to handle large amounts of genomes. Moreover, some of such approaches, for example, like CX1, is limited to short sequencing rates, but not genomes. And now, let me introduce some key points of our method. deBWT is a non-incremental approach. Mainly, it's based on 2 key techniques. One is the suffix comparison in different graph-based framework, and the other one is reduced BWT construction through k-means partition. Let me introduce the first one first. The deep graph of a given genome is a directed graph. The vertices are all the distinctive chimeras of the input sequence, and the edges are the connections of any two chimeras which have k-1 overlapping. In deep graph, the non-branch paths are usually called unipaths or unitakes. Here, a non-branch path indicates a path that the linear vertices are the single-in and single-out vertices. Moreover, it is also worth noting that a unipath can have more than one copies in the input genome. With this definition, the input genome can be represented as a permutation of the unipath. For example, in this case, the input can be represented as a permutation of U1, U2, U4, U2, and U3. Thus, any suffix of the input can be represented as a substring of a unipath plus the order list of unipaths. For example, the suffix starting at the position 4 can be as the substring of U1 and followed by U2, U4, U2, and U3. We call this the unipath representation of suffix. And in deBWT, the suffixes are compared using this representation. And given two suffixes, we have the following observations: that if the first chimeras of the two suffixes are different, okay, their order can be immediately determined. And if they are different, they must start from the same vertex of the different graph. Then, for two suffixes with identical initial chimeras, their order cannot be determined until they traverse different branches of different branch. For example, for the two suffixes starting at the blue vertex, their order cannot be determined by only considering the unipath the blue vertex is in. So we need to consider the next unipath, that whether the two suffixes go to the different branches. In this case, they go to the same unipath, I mean the red one, so the comparison need to go on. And at the end of second pass, the two suffixes go to a different branch. In this case, the green one and the purple one. This means that the next characters to be compared must be different, and their order can be determined. And now we found that the order of two suffixes with the same initial chimeras can be determined by one comparing the unipaths recorded in their unipath representations. However, we still need a fast way to do the comparison. Considering the definition of the unipath, it's easy to find that from the same end vertex of the unipath, it only needs to investigate one character to check whether two suffixes go to different branches. And if they go to different branches, their order can be determined. Otherwise, it also does not need to consider unipath, as it is the same again. So in one sentence, it needs only to compare one character for each of the unipaths. The character to be compared are the case characters of the unipath along the unipath representation of the suffixes. So to facilitate the comparison, we designed a new data structure called the Bruijn-branch encoding. Bruijn-branch encoding is a concatenation of these special characters, I mean the case characters of the unipath along the unipath representation of the whole input sequence. With this data structure, a projection suffix can be defined for any given suffix. Here, projection suffix means for a suffix starting, for example, a suffix starting at position I. Its projection suffix is the suffix of the string of Bruijn-branch encoding from here. Starting at phi-I. Here, phi-I is the character of the string of Bruijn-branch encoding, which corresponding to the case character of the unipath next to the position I. With these data structures, I mean with the Bruijn-branch encoding and the projection suffix, the comparison of two suffixes with the same initial chimeras can be done by directly comparing their projection suffixes, and it's much easier. And the second technique is the reduced construction through chimeral partition. It is easy to observe that if there are DG distinctive chimeras in the input sequence, The BWT of the input sequence can be partitioned into DG plus K parts. And all the suffixes with the same initial k-mers are in the same part. I mean, for example, all the suffixes initial with the k-mer 1 must be in the same block. And each of the k-mers correspond to a specific vertex of the different graph. With this definition, we have an interesting fact that if a part k-mer corresponding to a single unit vertex, according to the definition of BWT, all the BWT charts in this part must be the same. That's easy to justify with the different graph, as all the previous characters must be the first character of this vertex, so it is the same. Thus, if we have all the k-mers of the input sequence, and the k-mer partition of the BWT can be easily constructed by sorting the k-mers. And with a sorted k-mer table, there are at most UG unsolved problem. I mean, UG is the total number of the unit paths. So based on these two techniques, we developed two. Mainly, the deBWT construct BWT for the input sequence in 3 steps. Here, I briefly give the 3 steps, and please refer to the paper for the details, because there is some details. In the first step, deBWT employs a k-mer counting tool to get all the k-mers and their numbers of copies. Here, in current version, we use the jellyfish to do the k-mer counting. And then deBWT constructs and analyzes the different graph by k-mer sorting, and extract the multiple in and multiple out k-mers. And then in the second step, deBWT indexes the multiple in and out k-mers and generate the Bruijn-branch encode and the projection suffix simultaneously by scanning the input sequence. And in the third step, deBWT is sorting the projection suffixes directly to determine the BWT parts corresponding to the multiple in k-mers. Additionally, after these 3 steps, deBWT also handles some short suffixes, for example, suffixes shorter than k characters, and merging all the intermediate paths to accomplish the construction. And here are some benchmarking results. We use the dataset. We use 3 datasets to benchmark deBWT. Each is around 30 gigabase pairs. One is 10 in silico human genomes. Each is generated by including the variance of a 1,000 genome sample. And the second one is 3,000 assembled contigs. Because currently many sequencing rates are still assembled into contigs but not genomes. And the last dataset consists of 8 primary genomes from UCSC genome. And all the benchmarking was implemented on a server with 32 CPU cores and 1 terabyte RAM. The construction time of deBWT is shown in the figure. And we also compare the deBWT with ParaBWT and the RoBWT2. Mainly, deBWT is overall faster than ParaBWT. However, they are comparable. But we further found that mainly over half of execution time, deBWT was converting the output of jellyfish file to an interpretable format. Excluding this time, I mean the output of jellyfish can be, if the output of jellyfish can be directly read, deBWT can be 5 times faster than ParaBWT on human genomes and human contigs. This is the time course of the various steps of deBWT. You can see the time of file conversion. It is worth noting that the time of sorting the projection suffix is quite short. This is beneficial, as the fastest problem in BWT construction is well solved. And for other steps, it only needs to cope with integers, so it's much easier to solve. This is the time of deBWT in various number of threads. We also compare this with ParaBWT. It can be observed that when using more than 8 threads, the speed of ParaBWT does not change much. This could be due to it is an incremental method, and it's hard to scalable parallel computing. However, deBWT still has good speed up using many threads. This is because that deBWT is non-incremental, and most of the steps are easy to parallelize. However, there is still some processing in deBWT being serially operated. Most of them are intensive steps and quite slow. And we also tried various camera size. I mean, K equals to 19, 23, 27, and 31. It is observed that from 23 to 31, the speed is quite close. It's like due to that, the moderate long cameras may have similar ability to span short repeats, so that the structure of the generated different graph does not change much. However, when K is very small, the unit path will be shorter, and they will have more copies. Then the different branch encoding will be longer. Meanwhile, there will be more projection suffix to be handled. At last, we did some statistics on the 10 in silico genomes. The number of the multiple in cameras indicates the number of subproblems to be solved, which is 3 orders less than that of the whole length of the input. And the number of the copies of the multiple out cameras equals to the length of the different branch encoding. This is 1 order short than the whole input. This is also beneficial, as it is one of the main reasons to speed up the comparison of the suffix with long common prefix. And the method could be further improved in several aspects. Firstly, in the current version of deBWT, the comparison of the projection suffix is quite straightforward. I mean, directly compare the projection suffix. More advanced techniques, for example, different cover sample or something else, may further improve it. And secondly, the time cost of file conversion is a problem. It's a non-negligible problem. It's very beneficial to reduce the practical running time if other camera counting tools with the interpretable output format can apply. And thirdly, the construction and the analysis of different graphs relies on the sorting of the cameras. This is still not cost-effective. This should be also better implemented to further reduce the cost. And herein, we propose a new incremental BW construction approach, deBWT. The work is mainly led by Professor Yadong Wang, the director of Center for Barometrics, Harbin Institute of Technology, and co-worked by me and our student, Mr. Dixian Zhu. I also wanted to thank my colleagues in our new barometrics startup, Nebrogene, for their very helpful work. And deBWT is available at GitHub. You may scan the QR code to access. And give me 1 minute. There's a small ad, sorry. And we are recruiting. I mean, we are establishing a new barometrics institute in Harbin Institute of Technology. Harbin Institute of Technology is one of the top universities in China, and we plan to recruit 5 to 10 faculties in the next 2 or 3 years. The PhDs and postdocs in barometrics from highly reputed universities are welcome. And please contact me or direct to Professor Yadong Wang for details via the email. Thank you very much for attention. And